Hello and welcome to this session on form and function, how we can make sense of life by looking at protein structure. This video is a prelude to a live session that I'll be doing that will go into details about how we get protein structure. But before we get there, I'm going to talk to you about why protein structure is important. So my name is Dr. Tony Blake. I work at the University of East Anglia as a lecturer in biochemistry. I was trained as a biochemist and more specifically as a structural biologist. And because of that, I have a real appreciation for protein structure, why it's important, but also how beautiful it is. So let me talk to you about that. So proteins themselves are very important. I'll talk about some uses and why they're important in a bit. But throughout this video, what I'm going to talk about what proteins are and what they do. I'm going to talk about why the structure of proteins is important and why it's important for us to solve those structures and find what they are. And then I'll talk about how we got to our current level of understanding, and I'll finish off by going into some details about what I'll talk about in the live session. So I want to start off with this quote. So this quote comes from a book called The Science of Life that was written by H.G. Wells. So H.G. Wells is the same author who wrote War of the Worlds, The Invisible Man, The Time Machine, lots of fiction that was in the early 20th century. But he also wrote an actual biology textbook, and this was back in 1934. So in the section of the book where he's talking about nutrition and what we eat, he starts to talk about proteins after he's already talked about carbohydrates and fats. So he said that proteins, like fats and carbohydrates, can be burnt as fuel, and a large proportion of the proteins that we eat are used in this way. But they have another and more essential role, one which they alone can perform. For the actual fabric of the living cell is built largely of proteins. So not only can they be burned by our engines, but they are the steel from which these engines are made. I would actually go a step further than that, and I would say not only are the engines made of the steel, but all of the cogs, all of the control systems, everything that makes the machine able to work is also made of protein. A few sentences later, H.G. Wells says that any of the three kinds of foodstuffs will serve as an energy source, but protein is the only one that can be used for body building, for growth, and for keeping the tissue machines in repair. So let's have a look at that. So while we say that we are made of protein, so it's the steel that our biological machines are made from, it's actually about 20% of our mass. Most of our mass is water, then the next highest mass is protein. If we look at an actual cell, then we see the walls of the cell are made from lipids. They're not made from protein, although there is a lot of proteins in those. So if we look here at the cell itself, that's lipids. All of these parts inside, so the mitochondrion, that is lipid walls. The nucleus has lipid walls, but all of these lipid walls are also full of protein. And the parts that aren't enclosed by a wall, parts like this here, this is the cytoplasm, and while it's often drawn very plain, and you might think of it as just being some proteins floating about, that's not actually the case. If we take a good look, so a human cell is about 20 micrometers big. If we zoom right in to 100 nanometers, so that's 200 times we've zoomed in, then this is an artist's impression of what it might actually look like, with all of these things, with the green, the blue, and the red, all being proteins. So you can see that the cell is packed with proteins. These proteins are all interacting. Some of them are carrying out chemical reactions, others are transporting things or switching things on and off, lots of different functions. But before we talk about what proteins can do, what actually is a protein? So a protein is a biological macromolecule. This means it's a large molecule that's been synthesized by a biological system. And you can think of proteins as being a bit like beads on a string. All proteins are made from chains, and those chains are, like I said, beads on a string. Each of these beads is from a type of chemical called an amino acid. There are 20 different types of amino acid, and each of the 20 types has a slightly different side chain. So the backbone of the chain, this part here, will all link together to give us our beads on the chain, and then this part will be different depending on which amino acid it is. So here, for example, we've got 4 different amino acids. Each of those 4 have got a different chemical group pushing off to the side, which gives them different properties. Now that's all well and good, but just as a chain itself, a protein wouldn't work. For a protein to work correctly, it needs to fold into a 3-dimensional structure. So rather than looking like a bead on a chain, a protein looks something more like this. This is what we would call a space-filled model. So in this case, every atom is represented by a ball. So you can see all of those balls interacting with each other to make a large 3-dimensional structure. This structure will have a very specific shape and a very specific function. So let's now talk about those functions. So one that you may have heard of, you probably have heard of, is enzymes. So enzymes are biological catalysts. They're proteins that help a reaction to take place. Most of the reactions that we have inside our cells, inside the cells of all organisms, are proteins. So for example here we've got the citric acid cycle, sometimes called TCA cycle or the Krebs cycle. So if we have a look, then we can see that all of these things that I've highlighted, drawn circles around, they're all proteins that are acting as enzymes. So they're all allowing a chemical reaction to take place by lowering the energy barrier. Other things proteins do, so when we breathe in oxygen, we take it in through our lungs. It then needs to be transported through our blood vessels in red blood cells. Once it gets to tissues, the oxygen is released into the tissues. CO2 is produced, and that is released back into the blood cells, which travels back to the lungs and we breathe it out. But the protein that does this is something called hemoglobin, and again, that is a protein. Hemoglobin contains these oxygen-binding heme chemicals in it, which have got iron in. And because they've got iron, that's what makes it red, and that's what gives our red blood cells the red colour. So we're only able to transport oxygen through a protein. A lot of the structure that we have in our bodies comes from proteins as well. So for example, we have collagen that you find out kind of filling in your cheeks, filling in your lips. Also, your tendons are made of collagen, very strong, very long. And your hair and your nails are made from a protein called keratin. In both these cases, you can see that these are coils of coils. And this coil of coils gives kind of like a rope structure, which is all fastened together to give a lot of strength. Also, movement is caused by proteins. So what you can see here in the molecular diagram is myosin interacting with actin. And what we've actually got is just a few nanometers of movement. So that's billionth of a meter happening. And as this few nanometers shifts, we have millions and millions of these in line with each other, and that lets us get our macro size movement. Our immune system is made up of proteins. This is what an antibody looks like, and an antibody needs to be able to recognize different antigens, so bacteria or viruses. So it has to have part of it that is very variable, because that variable part will recognize different pathogens. But then it also has to have another part which is constant, so that the body recognizes that as being part of the immune system. As well as recognizing pathogens, proteins can also do signaling. So for example, this is the protein insulin. Insulin is obviously very important in glucose metabolism. It switches on when you're going to start storing glucose or releasing glucose, and that itself is a protein. And many of the machines that are on the tops of our cells that recognize different signals are also proteins. So that's just a few of the different functions that we get from proteins, but why is the 3-dimensional structure so important? Well, so far I've talked about all of these functions, and for the function to happen, we have to have the right shape. Biology is all about shape, especially when we get down to the molecular level. So far I've shown you some structures that look like this one here, where it's like a ball model where all of the atoms are shown, but often that's not that useful. We often look instead at the tracing the backbone and the different structures that we get there. So we often get like a helical structure, or the different strands. We call these an alpha helix or a beta strand, which becomes a beta sheet. By looking at this, we can see the overall topology and the overall shape of a protein. If we can look at the protein shape, we can also see where the important amino acids are that are going to do important jobs. So those jobs might be binding to a partner protein, It might be signaling to another protein. It could be detecting a pathogen. Or with this one, for example, this is an enzyme. This is the enzyme chymotrypsin. And if we look closely at where the active site is, the part where the actual reaction happens, we can look at the orientation of the side chains of the amino acids. And because we can see what orientation that they're at, we can characterize how that reaction happens. If we can characterize how reactions happen or how proteins bind together, that gives us the opportunity to try and design small molecules that could either activate this or inhibit it, which means we can use it to create drugs. Also, sometimes protein folding, turning from the beads on a string into the three-dimensional structure, goes wrong. If it goes wrong, that can cause disease. So a couple of examples here are the cystic fibrosis transmembrane conductance regulator. This is a protein that sits in the lungs and it pumps chloride ions from one side to the other. If this is not pumping chloride ions correctly, then that means that you get a buildup of liquid. That buildup of liquid gives you too much mucus. Too much mucus builds up in the lungs, making it difficult to breathe. And this is what happens in cystic fibrosis. Another example is prion proteins. So you might have heard of BSE or mad cow disease. This links to something called human variant CJD that we see in humans. This is a neurodegenerative disease where you get swelling of the brain and ultimately death. In this case, it's a protein called the prion protein, which you find in neurons, so cells in the brain, that's misfolded. So it folds into a version called the scrapey version. So this is the correctly folded version, and this is the scrapey version. If an incorrectly folded version comes in contact with the correctly folded version, that can cause that to misfold as well. If that misfolds, it'll cause more misfolding, and so on. If you have too many of these misfolded, then they'll start to stick together, they'll aggregate, and if they aggregate, they'll form something called an amyloid fibril. And these amyloid fibrils can damage the cells in the brain. We see amyloid fibrils in a few different diseases, so things like Parkinson's disease, Alzheimer's disease, lots of different neurodegenerative diseases. So these are all caused by proteins misfolding, which shows why it's really important to understand how proteins fold correctly instead. So now I'm just briefly going to give you a little overview of how we got to where we are at now. To do this, I've created in a program called Padlet online a timeline of how we got to our knowledge about proteins as we are now, our knowledge of protein structures. So you can use the QR code to have a look at that if you want. I'm going to take a quick look now and talk you through some of it, but I'm not going to talk through it all. But I'm just going to talk about some of the key factors, some of the key experiments which have led to our knowledge of proteins now. And I'm going to skip straight to the 1930s. So I'm going to skip to this. This is William Asprey. William Asprey was a biophysicist, one of the earliest ones of those, who worked on a technique called X-ray crystallography. So this is using X-rays to try and look at very, very small structures. It had been used in the past when it was first invented to look at things like crystal structures of diamond, crystal structures of sodium chloride, very simple things. So he wanted to look at proteins, but it was very difficult to get proteins to crystallize. So instead he was looking at protein fibers, and the protein fiber he was looking at was keratin. So keratin, I said before, comes in hair, it comes in nails, it also comes in sheep's wool. So he took sheep's wool and he fired X-rays at the sheep's wool when it was either relaxed or when he stretched it. He found that when it was relaxed, then he got one signal. When he stretched it, the signal completely changed. So he said that the protein here has two different forms. It has the alpha form, which is the relaxed form, the short form, or the beta form, which was the extended form. A few years after that, so if we go forward to 1951, this is Linus Pauling. At this point, we still didn't have any really good data to show exactly what protein structures were. So Linus Pauling started to build models instead. It's almost like building things from Lego. So what Linus Pauling did was he looked at all of the data. So the data that was the fiber diffraction data, but also looked at all the rules that he knew about chemical bonding and about how much rotation there could be around a bond, things like that. So using that information, he put together this model, which he called the alpha helix. and that matched up with the data that Asprey had for his alpha form. He also suggested the beta form, which was our beta sheets and beta strands. And then we get into the late 50s. By the late 50s, the technology had come on, and it was possible, with enough protein, to get it to crystallize, to purify the protein, get it to crystallize, and then use X-rays to get data from this that we could use to make structures. So the first crystal structure was of something called myoglobin, which is something that stores oxygen in muscles. And then the next crystal structure was hemoglobin, which is used to transport oxygen to the muscles. So myoglobin and hemoglobin were the first two solved. So that was the start of crystallography. From here on, there was an explosion of structures found. But then in the 1970s, people started to experiment with different techniques. So in the 1970s, people looked at nuclear magnetic resonance. So by 1984, it was used to solve the first protein structure. This was trypsin protease inhibitor 2A. But it wasn't until 2017 when people used a third technique to solve protein structures. In 2017, people used cryo-electron microscopy to solve structures for the first time. But new techniques, new computational techniques, and new image processing techniques allowed protein structures to be solved. As we stand now, we're moving through into a new era where we can use things like AI, a program called AlphaFold, to try and predict what a protein structure might be. But I'll talk about that a bit more in the actual session. So solving protein structures is not easy. It takes a lot of steps, but the first thing that you need to do to get any protein structure is first of all obtain a large amount of protein, generally at a high concentration. Now, when I say large amount, I'm still talking about kind of milligrams, not up to the grams amount. A milligram of protein is a lot of protein. So once you've got some protein, you need to purify it. There are a lot of different techniques that we would use to purify proteins, and I'll talk about some of those in the live session. Next, you would set up experiments and then analyze with your method of choice, whether this is crystallography, NMR, or cryo-EM. Generally, it's not going to work the first time. In that case, you need more protein. To purify more protein, then use the information that you discovered the first time you did your experiments to try again, and it's an iterative process until you finally solve the structure. So as I've said, there's three main different techniques that can be used: X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy. All three of them will give you a protein structure, but they all have different strengths, different weaknesses. Some of them are easier to do than others, some more difficult, but often depends on the protein itself. So in the session, I'm going to talk in detail about these three techniques and the strengths and weaknesses. So what can you expect from the live session? So I'm going to start off by talking about where we get proteins from and how we purify them. So before the session, something I'd like you to think about is the fact that the first proteins that were purified were myoglobin and hemoglobin. So this is a protein that you find at high concentrations in muscle and a protein you find in high concentrations in blood. So what is it about these proteins that made them good targets to be the first proteins to have their structures solved? Then I'm going to talk about how we would purify protein. So just as you might purify any other chemicals, how you might separate one chemical from another, think about the different properties the chemicals have. So proteins are just big chemicals. Think about the different properties that they could have that you could potentially use to separate one from another. Then I'm going to talk about the three major experimental techniques that we use to solve structures. I'm going to talk about the advantages and the disadvantages, the strengths and weaknesses of each, and when you might choose one over the other. We're going to talk about whether there is a best technique and when it's better to use one. But from the little you might know, maybe you've tried to grow crystals yourself, either at school or from like a science kit. What have you found when you've done that? And does that tell you anything that might lead towards some of the strengths and weaknesses? So hopefully after thinking about those, we can have a bit of a discussion about it in the live session. I'll tell you all about these techniques, so I look forward to seeing you then.